Hello everyone, welcome to the activities walkthrough portion of this week's lab for the ecdytozoans. So we're going to get started with the nematodes. Nematodes, better known as roundworms. This is one particular example of a human parasitic roundworm. If you never want to look at spaghetti the same way again, Google more images of them. I did not want to because they're... Bleh. Anyway, they've got this tough outer cuticle as part of all of the ecdytozoans do, which results in roundworms having these whip-like motions. Let's move on to a slightly less disgusting one, which would be the vinegar eels. We would have been looking at these in lab. They're microscopic, super tiny, but you get the same general idea. They get this whip-like motion. They grow in vinegar, as the name implies. They consume mother vinegar. I think that's what it's called. Anyway, they grow in unpasteurized vinegar, and should be able to get a little video of them moving. Yep. So you can see that whip-like movement as this is under the microscope, and it's just, yeah. So that's what they do. Roundworms. This is Turbantrix, and they're just doing their thing. Moving on from there, we have Onychonophora. I don't have a species given for these ones. Again, they kind of fall into this living fossil category, very cryptic, live in jungly underbrush areas. Watch the video that's on the playlist on the YouTube channel if you want to see these guys in action. They've got slime cannons that basically excrete super glue, and then a giant knife that comes out of their face to dust. They're predatory, and they're just super, super cool. The Onychonophora, still not a lot known about them. They've just been around for literally hundreds of millions of years. Now we've got the tardigrades, or the tardigrades, the water bears. These are one of my favorite things we'd ever look at in lab. One, they're adorable. Two, they are probably the best candidate for something that would be able to survive interstellar space and see life on another planet, because they are really, really hard to kill. I mean, you can physically squish them, but they go into this process of cryptobiosis, where if environmental conditions are unfavorable, they basically just shut down their entire metabolism. It's been suggested they might be able to live in there for at least 100 years. So they'll just wake up when the time is right. You'll see cryptobiosis come up again. However, make sure you watch the video that we made of the tardigrades in the lab. You can see them doing all the little tardigrade stuff. But because they are the ecdytozoans, they have to shed or go through ecdysis and shed that outer cuticle in order to get bigger. And you can see that here. And what happens is they have to shed that in order to get bigger, and then you're left with these little remains that look like little ghost tardigrades floating around. So make sure you watch the video on the playlist. You can only get to it through the link. Again, that's graded, so make sure you watch that, because I would have been doing a lot of time in the actual lab portion looking at them, because they're awesome. I mean, they can survive the vacuum of space. Moving on from there, we've got the arthropods. Arthropoda, like I said in the lecture, it means jointed appendages. So Now they've got these hardened cuticles on the outside, so getting more of like an exoskeleton. Massive grouping of organisms, and it's just huge. We're talking hundreds, I think millions of species. So yeah, moving on from there, we got some of the subcategories of arthropods. The Merostomatas are considered some of the most basal. Fossil records show them going back a very long time. Things to remember about them: their apomorphies would be the book gills and the telson. Right, book gills are going to be on the underside here, and the telson is this spine-like tail piece. It is not a tail, and you'll see why that makes a difference in a while. They've got, anyway, so that's the genus and species right there. They are primarily predators of benthic worms, so annelids, things you find in the ocean, tube worms and whatnot. And they can, if we ever see them in the lab, we get live ones sometimes, they'll curl on the bottom and they will bury themselves, so they'll just kind of burrow into the sand and they kind of move along. However, they can also swim. So make sure you get a good look about how these things are set up, because I think that might come up on the next exam. But you can see they got, the cheliceratas, they got these book gills and all that good stuff. They got some eyes on the top of their heads. Mouth is down here. So this is what they look like. They can be very tiny or they can be very, very large. And they can walk and they can also swim. So here's just a little of what a tiny horseshoe crab looks like when it's swimming, and then when they're walking along the bottom. This has happened to be a video I pulled from YouTube. Guy had one in his tanks, but we've had them in the past, and they do just this, and it's really cool. And then if they're going to go hunting for their benthic marine worms, they're going to be doing their thing on the bottom. Moving on from there, the arachnids, the ones we would look at the most in the lab, because we have them there, would be the spiders, the scorpions, but this also includes things like ticks. Make sure you're aware of what's going to unite all of them together. So we've got two body parts, cephalothorax and the abdomen. Right. This happens to be the garden spider that you would have been looking at. We got preserved ones of these in the lab. You would have been looking at them, so I got this one. So ventral view and dorsal view. So sorry, ventral and dorsal. So looking at it from the top down, from the bottom up. They have the chelicerae. They have the chelicerae, right? That's why they're in the big group of the cheliceratas. For them, they're a lot of times been modified into fangs and little pedipalps that help them push food into their mouths. And they have these 4 pair of walking legs, right? 8 legs. Spiders are known for them. So that's what makes them special, right? It's a real shame. Susan would have loved to see you guys. Susan is our desert tarantula. She is at least 21 years old. So she is super awesome to handle because she's very, very slow and not scary at all. But I will pass on everyone's, I'm sure, undying love for Susan when I see her next. So we got some diagrams for you to look at. So just make sure you can identify where the spinnerets are. So that's an apomorphy for the spiders, or sorry, the arachnids, spiders that you're going to want to be able to identify. You got the pedipalps. So you got these 4 walking appendages, 4 pairs. You get 8 of those. You've got pedipalps here, right? And then you've got fangs, which, if they're venomous, they use them. If not, they just use it to, bite things. The book lungs, you can see, are down here, right? No, not visible from the top, only from the underside. And then just some of the other, cephalothorax, your head and chest are together, and then your abdomen down here. All good stuff to know what is going on with the spiders. Scorpions. Scorpions are awesome. So moving on from there, right? Things that make scorpions unique, their telson. If you remember, we looked at the telson on the horseshoe crabs, their Merostomatas. Theirs has been modified into a stinger, and their pedipalps have been modified into their pincers. So we've got this telson that's modified into the stinger, right? And then its pedipalps are modified into these pincers. Makes them awesome. They still have the 4 pairs of walking legs, makes them special. Part of the grouped up there with their spider counterparts. But what also makes them super awesome is, okay, so before we get to what makes scorpions super awesome, here are some of the local scorpions that can be found in our region. We got the Arizona hairy scorpion. It is called the Arizona, but our small pockets are found in our region of Southern California. So be on the lookout for them. We have got the bark scorpion. Pay attention to this one. It is North America's most venomous scorpion. Probably won't kill you, but incredibly painful. Might kill your dog. They, like all the scorpions, they're predatory. They're great at hunting things at night. And so like scorpions are good to have around. Just be cognizant of where they are. And then we've got the striped-tailed scorpion. As you can see, not overly creative with its name. Its tail has stripes. Now, you may be asking yourself, how can I avoid scorpions, or how can I find scorpions because they're so awesome? Well, the answer is they fluoresce in UV light. Okay, so they have excellent camouflage, very hard for us to see them. They're nocturnal predators, so they're out at nighttime when we can't see very well anyhow, and they need to, hide from everything else that lives at night as well. But if you were to walk the desert with a UV flashlight, you would see something like this if you were to come across a scorpion. Now, what is that? So pay attention to that. So here's a little video. You can find these online. Tell me if you can see the scorpion. Probably not, because they're super well camouflaged. All right? Did you see it? No? Okay. Let's pop on the UV light, and bam, right there. They are that easy to see with the UV light. We're not entirely certain why they fluoresce under UV light. Obviously, it's not a natural condition for things to be walking around with a UV flashlight. Some suspect that it helps them see at nighttime because they'll always be fluorescing. It's just that in sunlight and regular light, all the white light drowns out what we perceive as the UV. But when we got this concentrated version of it, but long story short, we don't know exactly why they do it, but they do, and it is super cool. Moving on, we got the crustaceans. All right, this is a signal crayfish. So you would have been looking at these. We had a whole bucket of them for you to chop up, to look at in lab, already preserved and dead. This is a freshwater crustacean. Things to remember about the crustaceans: stalked eyes. All right, you can see those here. They have 2 pairs of antennae. Right, so we've got a big set, and then we have a smaller set, which is kind of hard to see there, but I think I uploaded a picture for you guys to look at. Right. And if it looks familiar, it's probably because it looks just like, a regular lobster. Why can't that erase? So let's see. Did I get a picture there? So, okay. You're supposed to rip off the carapace. I'm trying to work through the activities of what they looked for you to look at. So they still have this telson, right? But it has been modified into a big swimming tail. They have got several sets of walking legs. They're chelisopods, right? They got these big claws, and they have the rostrum, and they've got, you can kind of see it. They got these, okay, they got those compound stalked eyes, and they've got these little spikes right above their eyes. Helps protect those super delicate structures. And they've got the antennae, 2 sets of antennae. The large one for, feeling around stuff, and the smaller ones act as kind of chemoreceptors. Not the same thing as a nose, but similar, and helps, pull out chemical signals from the water. Yeah, so again, this is the signal crayfish. There's the name for you guys. Now, off of our coast, we got the California spiny lobster. These are super awesome. They can get really, really big, as you can see here. This one was locally known as Larry the Lobster, and he was safely returned. We always have some. He was returned to the ocean that he was returned, so he can get even bigger. We always had a couple in the tanks. So the spiny lobsters are different than the main lobsters who have the big claws because they don't have these big protruding petapods like the main lobsters do. However, they have these two sets of antennae. Right? So you can see those there. They got those big ones, and then they got the smaller ones. Right? Again, that is one of the big ones, and that is the smaller ones. If you can see, it's bifurcated, so it's two, but it's actually got four little ends here. So two pair of antennae. But they use these big ones for feeling around, but they can also use them for really well for defense. If you were to put your hands on them, which we would always have these molting ones, we have these molted exoskeletons of them in the lab for you to touch. They are very spiky, going one direction. Right? So they're like kind of not barbed, but sharp, and they can cut you if they whip you with them, which is why I never pulled them out of the tank. Okay, next we're going to look at barnacles, and we're going to look at two orders of barnacles. They are under the crustaceans, right? So, but the Cecilia or the acorn barnacles, and the Pendiculata, also called gooseneck barnacles, these are orders, right? So they are not genuses or species. This is just a massive grouping, right? This is a particular genus and species, and so is this. There's nice little examples of them that can be found in our local area. But there are crustaceans, there are filter feeders, and what you want to pay attention to when you're looking at them are, so if you've ever been out of the tide pools, barnacles are everywhere, right? These are the ones that are really common to see, right? Acorn barnacles, named so because they look like about the size shape of an acorn. All right, but Cecilia, they're sessile, right? They are attached, they're not going anywhere. And what they do, right, they are filter feeders. So they've got, they're just attached to the rock, and they got these little cirri that are coming out. Can you see? No, you can't see that. There we go. Right here, right? So those are going to extend into the water column, and they're just going to be pulling anything they can out, right? And to be grabbing onto stuff. So this is a Balanus glandula. Okay, so just a classic looking acorn barnacle. Here's a nice cross section. They are hermaphroditic, so as you can see, it's got both a penis and the egg mass in there, right? And it's just going to keep pulling stuff out and trying to feed itself. And now, if we look at the next one, we've got the gooseneck barnacles, called such because, well, cool story. They used to think, before we knew anything really about science, about spontaneous generation, they thought that these became geese. But we kept the name because they look like little goosenecks. Anyway, not relevant at all. So these are the other types of barnacles of the really big orders of barnacles. Right? This is the polyceps. And if you were to give them a bit of a squeeze, you can see their cirri come out. Again, you can kind of see it here. Right? They do still the filter feeding. But unlike the acorn barnacles, which have that little protective shell that they kind of hide and close themselves into, this is what they look like on the rocks. Right? So these aren't in these massive wave-exposed areas like acorn barnacles are. They're going to be in a little more wave-protected, so not quite as rough. Right? Not that big splash, so with all that tremendous wave energy, because they can still, they're moving around a little bit. I mean, they're sessile, they're still attached to the substrate, but they can't be, they're not as hardy as the other ones are. All right, so they still have those cirri, right? Then they get the name from this peduncle, right? Just this little kind of like a pedestal that will do what they do. All right, we got their cirri still, and I've got a juvenile barnacle coming off the side here. All right, so they're just kind of budding off, and they'll do their thing there. Now, what makes barnacles super interesting, think about this. They're sessile, they are attached, and they can't move, obviously, but they do reproduce sexually, and most of the time, as far as I know, they're not broadcast spawners. So they have evolved some of the longest penises in the animal kingdom compared to body size. Some of them have penises that are up to 8 times as long as their bodies are. Because if you're attached to a rock here and you need to mate with somebody over here, you will send out your groping penis until you find somebody to mate with, and then you will do your thing. That's kind of cool. Moving on. Berry shrimp or brine shrimp. Okay, Artemia is yours. You were supposed to be looking at in your lab manuals. So some of you might have had sea monkeys as kids, all right? But those are, so like the tardigrades, these can go into a state of cryptobiosis with their eggs. And so somebody figured out in the 50s they can collect those eggs, and then you put them in a little packet, you put them in water, and they will just grow, right? But that's what those are. So Artemia silenia. So you might have heard of fairy shrimp or brine shrimp as a big point of contention in land conservation in the areas. I urge you to watch the video that's part of the playlist for these, but you'll get these really arid desert regions that have very ephemeral ponds and lakes that'll spring up in the wet season and then dry. And so you'll be in an inland, dry lake bed, waters come, and then you'll find berry shrimp in there, right? And then as soon as they dry out, they lay their eggs, the adults die, and then they go about their business. So they look really different, but you got the female here and you got the male here. So watch the video on the playlist about the fairy shrimp in the desert. It is super, super cool, right? Then they're just doing their thing. Daphnia, these are the water fleas that we've looked at least 3 times now. But this is where they officially come in, is they're part of the crustaceans. So these are, again, freshwater water fleas. Really cool. You can watch the heartbeat. And these are the ones that we had fight with the hydras. And usually the hydra's won, but sometimes the Daphnias won. But again, that's what you're supposed to be looking at for the Daphnia. They're just really little cool water fleas. Hexapodes. All right, Hexapodia, classic insects. Now, these next couple of slides were put together by the other TA, Eric, who went and we have a massive collection of stuff that we're big university that's been around for a very long time. So in our collection, he went through and got some really awesome examples of stuff that we have on hand. And I'll be showing you some of those in a second. But insect development, I'm not sure if some people might it's usually some confusion about what the different types of metamorphosis are. So we've got hemimetabolous and holometabolous. What you want to remember, complete metamorphosis, so you are completely different, and incomplete or half, all right, hemi, half. Right? So you got eggs, then become nymphs, then become adults. The nymphs look just like little, less developed versions of, nymphs look like little, less developed versions of the adults. Compare that to complete or holometabolous development. Think about it, you got like eggs, larvae, think of maggots and caterpillars and things like that. They go through a pupa and they metamorphosize all the way to an adult butterfly, all right? Caterpillars do not look like butterflies. Grubs do not look like beetles. So if you have that, you go from a little grub stage to an adult stage that are completely different, you go through complete metamorphosis. All right, first grouping we're going to be looking at is the Odontatas, right? So these are your dragonflies and damselflies. Same with all the hexapods. They've got all 6 of their legs attached to the thorax, right? Nothing is attached down here to their abdomen. Got 2 pairs of wings, right? Dragons, damselflies happen to have really big wings, really awesome. One of their traits here is that they have an aquatic stage of life, so their grubs, are not flying in the air, but they're swimming in water. Some of them can get really big, eat small fish, but then once they develop, they grow up, they leave, so they go from the water to the air, and I think that's pretty awesome. Again, large compound eyes, well-developed jaw, and they are voracious predators, right? Like it says here, they're high in the food chain in their larval and their adult stages. Next, we're going to look at the Orthopteras. This is going to be things like grasshoppers and crickets and katydids, cicadas, locusts. Right? Things to be noticing for them, they got these massive jumping legs. Okay, they also have wings, right? But they don't use them for consistent flight like you would see with the dragon and damselflies. They've got very complex mouth parts, right? That's why they can go chewing through whole fields of stuff. They can get really, really big. So the large African locusts, right? 15 centimeters, it's like 6 inches. If you're wondering the difference between a cricket, and a grasshopper, and a locust is, I'll tell you between a grasshopper and a locust, they're very, very similar in appearance. But the locusts are different because they have two behavioral states. They've got solitary states in which they behave almost indistinguishably from a grasshopper, and they got a gregarious state, which is when they swarm. And when they swarm, they become a plague, and that's the actual term for a plague of locusts is when you see these massive clouds, and they will, when the population density gets high enough, they will basically flip on swarm plague mode, and they will swarm across the countryside of wherever they're at and devour every edible piece of vegetation that they can until they run out of food or something else stops them. But it can get, it can be devastating for the entire ecosystem if these things swarm, and they do every once in a while. And there's a reason they're called a plague. Right? Plague of locusts is not just hyperbole. It's bad. The coleopterans, right? So these are your beetles. We have named over 400,000 of them, and they come in every size and shape and color out there. Some of the really cool ones that he got for you. So you've got these longhorn beetle species from Mexico. All right, look at that's all going on there. All right, you got rhinoceros beetles, aptly named. You've got, they still have these two pair of wings. First pair, so they're not as, first pair is not flying wings. They're more like the shell covering, and then they open those up, and then the second wings come out. They're hind wings, right? Which those can actually be used for flying. Just really, really cool things. Yeah, that's dung beetles and scarab beetles and big rhino beetles. I would recommend highly looking online. I don't think I put it on the playlist, but there is a time lapse of these great rhino beetles going through complete development. So you see them in this grub stage that gets massive, and then it turns into one of these. What is super, I'll get to it in a minute. The Hymenopteras, all right, 150,000 species, but these are things like your wasps and bees, ants, and things like that. Not all of them are going to have wings. Some do, some don't. But when they do have them, they're going to have these, two pairs, two mouth parts, thorax, abdomen, oftentimes this narrow little waist, right? But still, everything, all the legs are going to be attached to the thorax, nothing attached to the abdomen. This is a tarantula hawk wasp from Pepsis, is the genus, right? Come from Mission Trails. Said to be one of the most powerful, painful stings on the planet. I don't know that for certain. I don't want to find out either. And some of the other really cool ones. Anyway, look through some of the stuff that we got here. Super cool. Japanese hornet. I think I put it out on the other lecture slides. They can get big and terrifying. I'm just glad they're not bigger than they already are. The dipterias, all right, 125,000 species that have been described of flies. So, they've got one pair of wings, right? And these are going to be lots of different things to look at, all right? Right, so what here, that is still a fly. It has mimicked itself to look like one of the Hymenopteras, right? A lot of them don't have the ability to sting, but if they look like something that does, you get this mimicry going on. Then it's like, hey, remember this warning coloration, the aposematic coloring? Well, they're trying to mimic something that does have a threat to them, even though they are not. So it mimics a bee, saying, stay away from me, I'm dangerous, even though it is a harmless little fly. The Leodopterans, right? These are going to be your butterflies, moths, and those sort of things. Two big pairs of beautiful wings. You'll see the wings up close here in a second. Really cool thing that not only, so when they go through this complete metamorphosis, their entire external and internal structures change. But even though they basically turn to goo and then re-solidify, they maintain memories. There's an experiment done where they train the caterpillars to react to, I think it was a smell or a sound with a pain or fear response. And they determined that after they went through this metamorphosis, it came out as butterflies, they still remembered that response. So they had that learned behavior even after they became an adult. And there's a lot of examples of coevolution as well. So certain species of butterfly can only pollinate certain species of the Passiflora, so the passion flowers, and they've co-evolved together. So they're the only things that can be pollinated by them, and they're the only things that that particular butterfly sustains itself on. So this here is up close on a butterfly wing. Yeah, butterfly scales, right? So a lot of times you'll see these magnificent colors, but it's not actually a pigment. It's just the way that they're reflecting, and based on the physics of light, it can reflect it at different wavelengths, and you get some of these different colors. This tobacco hawk moth, it's just kind of cool looking, right? Moving on from there, now we get into the Myriapoda. We had the Hexapodas, now we're at the Myriapoda. Two classes that we will look at, right, are the Diplopodas and the Chilopodas, so the millipedes and the centipedes. But in general, Myriapoda, as its name implies, myriads of legs. Some of them have up to 750 legs, but not, I don't think that those are millipedes. Anyway, so we got a nice example here, scarlet millipede, and then a common or stone centipede. Go over here, centipede, millipede, and we'll look at them together. You're supposed to be able to identify the difference between the millipedes and the centipedes. So we're going to go over that real quick. All right, so millipedes, when you're looking at them, each segment has two pairs of legs. So you've got segment, 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 and then attached to there is going to be two pairs of legs. So we got two legs on this side and two legs on the other side, so two pairs. Centipede, each segment has one pair of legs. So you've got your segment, 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 and each one has got one leg, so one pair, right? So 2 legs per segment, 4 legs per segment. So we'll look at them in pairs. They both have one set of antennae, right? Larger set of antennae. The millipedes are typically going to have some shorter antennae. They both have a mouth, obviously. They both have a head. Now, another big difference is millipedes are primarily detritivores, and centipedes are mostly predatory, right? And because centipedes are predatory, they have venomous fangs. We'll cover it in just a second. Looking a little closer at one of the local millipedes we have in the area, so the Diplopodas, right? So they've got the Hiltonius, it's a genus. There's several different kinds of those. But again, detritivore, 2 pairs of legs per segment, 1 set of antennae. You can see there, these little short ones. They are endemic to our region, so these can be found all throughout Southern California, but they're detritivores, so just leave them be. They're doing their thing. They're just kind of breaking down organic material, and they're just cool. Chilopodas, looking at some of the centipedes, right? This is the house centipede, right? Can be found all over the world. Where you see people, you're going to find these guys, right? Locally found as well. 1 pair of legs per segment, right? So the segments aren't as readily identified just from this particular picture, but you know, 1, 1, 1, coming out to the sides. They are venomous and 1 set of antennae. So you can see the antennae here, all right? Antennae there. And then below that, we have got, if you look in there, you can kind of see that they've got these venomous fangs. And remember, they're venomous, not poisonous. If it bites you and you get sick, it's venomous. If you bite it, and you get sick, it's poisonous, right? So think like a poisoned apple versus a venomous centipede or a venomous snake. So get one set of antennae, one set of legs per segment, venomous fangs, difference between centipedes and your millipedes. So this comes to the end of all the stuff that we're going to be able to show you for this week's lab. Please make sure that you watch the videos on that playlist. Make sure you open up all the links and material that are for you guys on Blackboard. We used to do the stats tracking of those so I can see who's been opening them and who hasn't. That's easy points you're throwing away if you don't follow those links, guys. Please do that. Don't make this any harder than it has to be. You have 24 hours from the end of our scheduled lab time to do the lab write-up. Compared to last time, I'm looking for more details. Don't describe the entire orders or classes of organisms. Talk to me about individual genuses and species. Don't just describe crustaceans. Describe the spiny lobster. Don't describe hexapods. Talk about, be more specific. All right, that was way too broad last time. 24 hours, and if you have any questions, don't hesitate to email me or set up a Zoom conference. And yeah, so we're good to go. Thank you very much.